Hello students, welcome back to Digital Learning DCE. Myself Dr. Manjula. In this session, let us study about bacterial cell wall, gram-positive and gram-negative bacteria and other structures like nuclei, pilli and flagella. Let me begin with the introduction. Bacteria are microscopic, least differentiated primitive organisms which are present everywhere on the earth's surface where life can possible. They exhibit a typical prokaryotic cell structure which lacks true nucleus and also membrane-bound cell organelles. These organisms may be unicellular or multicellular. Based on their shell shape, they are of three types. Rod-shaped bacteria are known as bacilli, spherical-shaped bacteria are known as cocci, and helical-shaped bacteria are known as spiral bacteria. Based on mode of nutrition, bacteria are of two types, heterotrophic bacteria and autotrophic bacteria. Heterotrophic bacteria depend upon another organism to getting their nourishment. They may be saprophytes, parasites or symbionts. Whereas autotrophic bacteria, they are provided with special type of pigment called bacterial chlorophyll. Hence, they synthesize their own food. So, ultrastructural studies of bacteria reveal that a bacterial cell is provided with three important layers. The outer slimy layer, which is gelatinous in nature, is called as capsule. This slimy layer is followed by a middle layer called cell wall, and the innermost layer is called as cytoplasmic membrane. Let us study the bacterial cell wall structure in detail. The bacterial cell wall is a granular structure which is tough and rigid. It gives shape and rigidity to the bacterial cell, which varies in thickness from 50 to 100 angstrom. It mainly composed of three important substances like N-acetyl glucosamine, N-acetyl muramic acid and a polypeptide chain. Together form a layer called peptidoglycan layer, otherwise known as mucopeptide. In this diagram, we can observe the chemical composition of bacterial cell wall where N-acetyl muramic acid and N-acetyl glucosamine are arranged alternate to each other in a single direction, whereas a peptide chain runs in a crosswise manner. Along with these three major substances, there are also other additional substances found in case of bacterial cell wall like teichoic acid, protein polysaccharide, lipoproteins and as well as lipopolysaccharides. So, based on the staining technique developed by a bacteriologist Christian Graham in 1884, bacteria are categorized into two types, gram-positive bacteria and gram-negative bacteria. Gram-positive bacteria are bacteria which will remain purple in color followed by gram staining technique, whereas gram-negative bacteria appear pinkish in color after gram staining procedure. This differential staining is mainly due to differential presence of the cell wall in case of bacteria. So in case of gram-positive bacteria, there is a large amount of peptidoglycan layer which also lacks the outer lipid membrane. Whereas in case of gram-negative bacteria, the peptidoglycan layer is very thin and there is also the presence of both inner lipid membrane as well as an outer lipid membrane. This differential composition brings out the difference in gram-positive and gram-negative bacteria. As I already told, the bacteria which retain the primary stain, that is crystal violet, during the gram staining procedure, even after using the decolorizing agent alcohol, they are called as gram-positive bacteria, whereas the bacteria which are going to lose the primary stain crystal violet, and it retains the counter stain, that is safranin, which look pale pink in color. They are called as gram-negative bacteria. This differential reaction is mainly due to the differential amount of lipids and peptidoglycan layer in the bacterial cell wall. In case of gram-negative bacteria, they have high lipid content since there is both inner lipid layer and outer lipid layer are present along with a thin layer of peptidoglycan. Whereas in case of gram-positive bacteria, the peptidoglycan layer is very thick and it lacks the outer layer of lipid. During gram staining technique, since in case of gram-positive bacteria, the peptidoglycan layer is thick, when we use a decolorizing agent, that is alcohol, there is no outer lipid layer, so there is no leakage in this cell wall, hence the primary stain remain as such. After the gram staining technique, these gram-positive bacteria look purple in color, whereas in case of gram-negative bacteria, since there is relatively less amount of peptidoglycan and also outer lipid layer is present, when we add the decolorizing agent, this outer lipid layer dissolves and damage is taken there, which will start to leak the primary stain, that is crystal violet, when we use the decolorizing agent alcohol. When we add the counter stain, that is safranin, then it takes that particular stain and remains pale pink when we observe under light microscope. Hence, gram-positive bacteria are purple in color and gram-negative bacteria are pale pink in color, which is mainly due to the differential amount of lipids as well as the peptidoglycan layer. Then coming to the bacterial nucleoid. In bacteria, we can observe a special type of nuclear material, which lacks the proper nuclear envelope and also nucleus. In case of bacteria, the nuclear material is simply dispersed, simply floating inside the cytoplasm, which contain tightly packed DNA. This is otherwise known as nucleoid. So nucleoid can be defined as irregular shaped, tightly packed fibrillar DNA within the bacterial cell, which is otherwise known as the genetic material of bacteria. So this is a characteristic feature of prokaryotic organisms. That means incipient nuclei or primitive nuclei is present without the nucleolar membrane as well as nucleolus. Hence, the genetic material of bacteria is called as the nucleoid. Coming to the structure and function of bacterial pilli. In some case of gram-negative bacteria, we can observe small, thin, air-like structures which are proteinaceous in nature found on the surface of bacterial cell. These are referred to as pilli or fimbriae. The main function of pilli is to attach a bacterial cell to specific surfaces or to other bacterial cells. Along with this small hair-like structures, pilli, in some case of bacteria, there are special pilli are going to be formed during the process of conjugation. Those pilli are referred to as conjugation pilli or sex pilli, since they allow the transfer of DNA from the donor bacterial cell to the recipient bacterial cell. In this diagram, we can see the formation of sex pilli or conjugation pilli, where the transfer of genetic material from the donor cell to the recipient cell takes place through this conjugation tube. Hence, this pilli is known as sex pilli. So the main function of pilli is to attach the bacterial cell to the specific surface, whereas the sex pilli aids in transfer of DNA from the donor to the recipient. Coming to the structure and function of flagella. These flagella are long, thin, whip-like appendages which are also proteinaceous in nature that comes out from the surface of bacterial cell. So this is a long, thin, whip-like structure which comes out from the bacterial cell surface, which is made up of three important parts. The basal body. This basal body, it is inside the cell wall, which mainly made up of 9 pair of flagellin protein. Then there is a bent-like structure which arises from the basal body. This is known as hook, and hook extended to form a long whip-like structure that is known as a filament. The movement of the to and fro movement of this filament aids in locomotion of this bacteria. Hence, main function of flagella is to bring about the locomotion or movement in the bacterial cell. Along with that, sometimes they also act as sensory organs to detect the temperature and pH change in the medium which they grow. To sum up this session, as I told, bacterial cell wall is a granular structure which is tough and rigid. It gives particular shape to the bacteria and rigidity to the bacterial cell. And based on the Gram staining technique and the composition of cell wall, bacteria are of two types, Gram positive bacteria and Gram negative bacteria. Gram positive bacteria appear purple in color and Gram negative bacteria appear pale pink in color after Gram staining procedure, which is mainly due to the differential amount of peptidoglycan layer as well as the outer lipid membrane, which is lack in case of Gram positive bacteria and outer lipid membrane is present in case of Gram negative bacteria. The nucleoid is a genetic material which is otherwise known as the fibrillar DNA without the nuclear envelope and nucleolus. So hence this is a primitive nature which is a prokaryotic nature. The pilli are thin air-like appendages made up of protein found on the surface of the gram-negative bacterial cell, which generally helps in adhesion. Some special type of pilli which aids in transfer of genetic material are called as sex pilli, and the flagella are long, thin, whip-like appendages comes out from the bacterial cell surface, generally helps in locomotion or movement of bacterial cell. So for further details, you can refer the following references. With this, I am going to conclude the session. Thank you.